Gastric acid primarily acts to convert the inactive zymogen of pepsinogen into pepsin, an enzyme secreted from cheese cells in the stomach fundus. Pepsin breaks down proteins in a process known as proteolysis and also directly denatures peptides into smaller amino acid sequences due to its high acidity. It is mainly composed of hydrochloric acid secreted from parietal cells, but also contains potassium chloride and sodium chloride. This presentation will cover gastric acid formation and its regulation. Gastric acid formation begins with carbon dioxide that leaves the blood and enters parietal cells. Here it combines with water molecules and forms carbonic acid, which dissociates quickly to bicarbonate and protons, catalyzed by carbonic anhydrase. In the basolateral membrane, the bicarbonate ions are actively removed via an antiporter protein in exchange for chloride ions. In the apical membrane, there are ATP-dependent active proton pumps, which pump protons out of the cell and into the gastric lumen in exchange for potassium ions. The chloride ions and potassium ions move passively out proton channels and into the gastric lumen. Here they combine to form hydrochloric acid. The main gastric acid regulatory pathway is a binding hormone called gastrin. Gastrin is secreted by cheese cells and are secreted in response to proteins entering the stomach and temporarily increasing the pH. The gastrin travels in the blood to the parietal cells, where it binds to CCK receptors on the cell surface membrane. It stimulates an increase in proton potassium pumps to be inserted into the membrane. Gastrin also binds to ECL cells. Control is also neuronal via the parasympathetic vagus nerve. It secretes acetylcholine to bind to muscarinic receptors, further increasing the number of proton potassium pumps. Histamine is released from ECL cells and binds to H2 receptors, having an amplifying effect. If the pH in the lumen drops too low, the hormone somatostatin is secreted from D cells. It has an inhibitory effect on G cells, ECL cells, and parietal cells, ultimately decreasing gastric acid secretion.